Hello everyone. Let's study the chapter 8, Microbial Genetic and Genetic Engineering. The genetics is the subject to study the transfer of the inheritance and the heredity of living things from generation to another generation. So it explores the transmission of the biological features, traits from parents to offspring, and how the traits are expressed in the living organism and the structural function of the genetic materials. The genetic materials are the DNAs, right? Not DNA, and how DNA changes. So the genome is the concept to describe the total genetic materials of an organism. So actually, the genome means all the informations on the DNA and the chromosomes. Okay. So so there are there is another DNA materials such as the plasmid. Okay, so the inheritance information on the plasmid is also considered as the genome. Okay, and so for the eukaryotes, the genome means all the information in the entire DNA. For the virus, so since the virus has two types, right? One is the DNA virus, another is the RNA virus. So the genome means the information on the DNA, either DNA or RNA. So the chromosomes. Actually, chromosome is compounds of the packaged DNA. Okay, and packaged DNA. So that's double-strand DNA plus the histone protein and the other proteins related to regulating the DNA. Okay. So eukaryotic chromosome includes DNA. So the DNA actually, so the DNA is wounded around the histone protein. Histone protein is the series of histone proteins. So those proteins actually maintain and regulate DNA structure. So the cluster of DNA and histone protein, so actually is chromosome. Okay, so it is located in the nucleus. So in the mature cells, so it is diploid, means the cells have two copies of chromosomes, and in the gametes, so it's haploid, so there is only one copy. Okay, so this is a linear structure. For the bacteria, so the bacteria's DNA is shorter and simpler. Okay, so the DNA can be condensed and packed with histone-like proteins. Okay, so those proteins also maintain the DNA structure. Some bacteria have the circular chromosome. Okay, and so the circular chromosome is very stable. So so those type of DNA may not need too many histone-like proteins. So for the genes, So we have structured genes. Okay, so structured genes. So those the genes code for the proteins, because all the proteins are used to build our body. Okay, and make the enzyme for the metabolism. Okay, so this is called structure genes. And also some genes code for the RNA machinery. Okay, so they are used to make the ribosome to produce protein. And regulate so the regulatory genes control, so those genes actually controls the gene expression. Okay, so they can make some proteins, so those proteins can control the gene expression. Genotype is all the gene type. Okay, and so this is the genetic makeup. Okay, so if all the information of the genome is expressed to make the proteins, make the features of the living organism, so the features and the traits is called phenotypic. Okay, the phenotype. So it's the expression of the trait. So we can see here. So this is so this is eukaryocytes. Okay, eukaryocyte has the nucleus. So chromosome is in the inside of the nucleus. So this is mitochondria. So in fungi, some fungi have the plasmids. So plasmids are used for transformation of the DNA information. So the prekaryocytes, the proteocytes don't have the nucleus, so they may have the circular chromosome. Okay, circular chromosome actually is simple and stable. Okay, virus, so some virus contain the DNA. Okay, some virus contain RNA. So the DNA code, DNA uses 4 nucleotides to make the genetic information. Okay, so so they are called they are called base unit. Okay, base unit. And so the DNA also has the phosphate and deoxyribose and nitrogenous base. Okay, nitrogenous base actually can be made for the nucleotide. So the nucleotide can bind each other's. So the nucleotide can bind with sugar phosphate backbone with the covalent bonds. Okay, so the covalent bonds is very strong. So in the backbones, so each sugar attached to repetitive two phosphate. Okay, so one can be attached to a number of 5 carbon, and the other can be attached to the 3 carbon. Okay, so so the attachment to the 5 carbon or the 3 carbon makes the direction of the DNA strand. Okay, so we can count in the 5 so the combination to the 5 carbon, either 5 carbon or 3 carbon, to make the direction for the 2 DNA strands. Okay, because DNA has 2 strands. One strand is the direction from 5 to 6 to 3. Okay, another is also from 5 to 3. However, so the directions are opposite to each other. So the nitrogenous base attached at the one position of the sugar. Okay, with the covalent bonds. So that means so it's attached So it so it so the so the linkage between the base and the sugar is very strong. And then the purine and pyrimidine join with base and using the hydrogen bond. Hydrogen bond is very weak, so that's why so the purine and the pyrimidine can be detached, unzipped. Okay, because the hydrogen bond is weak, so they can be so they can be break down. Okay, so they can be broken down and easily unzipped. Okay, unzipped to expose the sequence of the sequence of the base, because sequence of the base actually is the genetic information, and then so the DNA strand can be either replicated or the transcripted. Actually, so normally, so the nitrogenous base are each other. Okay, so for example, adenine is paired with the thymine. Okay, guanine is paired with the cytosine. Okay, like this diagram. Okay, so this is the double strand backbone. Okay, so we can see. So this is the phosphate group. So this is the deoxyribose. Okay. Mm. So we can see here, here is the 3 points. Okay, so here, so for this one, so the direction is from this 5 to 6. Okay, however, for this string, so the 5 point is here, 3 point is here, so the direction is this way. Okay, so they are so the direction are opposite to each other. Okay. So the base pair, so this is guanine, this is cytosine. So they are so these two base are paired each other. Okay. So the structure of the DNA double strand is the helix. Okay, the helix. So this is called anti-parallel because we can see the direction of 5 to 3 is opposite to each other. So this is called anti-parallel. Okay, because they are run in the opposite direction to each other. Since uh, since the bond between the carbon and the deoxyribose and the phosphate, so those chemical bonds are the covalent bonds. So the covalent bonds are very strong. So actually, so this structure is very strong. Okay. So for example, here, so one is reference five. So if we read the two strings from one direction, actually one is from 5 to 3, another is from 3 to 5. Uh, so so based on different reading fashions, we can get a different reading. Okay. However, so actually, if the double strands are separated, so the one strand can be used as the template for the replication and the transcription. So the enzyme always reads the information from the DNA strand from 5 to 3 direction. Okay, so this is very important for the DNA direction. Okay. Now, DNA replication means the DNA duplication. Okay. So this is called a semi-conservative replication. So the enzyme will so the a cluster of enzymes will carefully uh, read the information from the DNA string. Uh, so so the enzyme can separate the string. Okay, so the separate the string. So this is called the helices. Okay, so they can separate the string and expose the base sequence, and then the polymerase. So this is another enzyme. Uh, can 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 make the duplication of the DNA string. So the new new DNA string is called a daughter. Uh, it's called daughter string. So so all the molecules on the daughter string actually is identical to the parents. Okay, so this is actually is identical. Identical means it is exactly same. Okay, so this is why it's called semi-conservative. Okay, because it make one is because it's duplicated, so all the information are exactly same. So this means uh, one old DNA strand can be duplicated with one new DNA strand. Okay. However, all the chemical components and all the base informations are exactly same. Okay. So this means it's not complete. It's not complete new. Okay. Because the new DNA double strand consists of old one piece of old string and one piece of new string. Okay, so that's why it's called a semi-conservative replication. So we can see here, so those are the enzymes used for the replication. Helices is for the unzipping, okay, to detach the double strand. So the pyramid is to synthesize the polymer uh, to make the location for the replication, and all these primers are used for the for the duplication of the DNA strand, and ligase is for link the synthesized DNA fragment. So we can see in this diagram, okay, so the helix will unzip the DNA double strand. Okay, so all the base sequence will be exposed, and then the polymerase can so first the primers will make the primers and to set up the location for the synthesize, right? And then the polymerase will duplicate read the information and duplicate another DNA string. Okay. So here, since we can see, so this is completed from 5 to 3. So actually, so the polymerase can read this continuously. However, for this actually, uh, so here is, so this is 5 to 3. So that means if we read here, so this is 3 to 5. Okay, 3 to 5. So that means for this string actually, so it need so the polymer can make duplication one fragment and one fragment, and then so they need a ligase to link the gap between the different fragments. Okay, and then so the helices can continue unzip the DNA strand, then so the polymerase can continue replicate until all the whole DNA strand are replicated. So the replication actually is very so the replication is processed very quickly. Okay, some bacteria can make at 750 base per second. Okay, so this is very fast. Okay, so the polymerase one actually removes the RNA polymers and replace them with the DNA. Okay, and the ligase uh, will link the different fragments and add the nucleotides to fill in the gap to link the fragment together. Okay. Actually, during the replication, so some mistakes may happen. Actually, it is widely common. Okay, so about 10 uh, 10 ninths and 10 tenths times base pair can have the mistakes. Okay, so those mistakes can be corrected by the polymerase 3. Okay. Also, so the replication can happen from different positions simultaneously. That's why, for example, in this diagram, you can see the replication is started from two positions simultaneously, so they can make a double loop. Okay, and then so the direction is from here to here. Okay, okay. So when they reach to the final point, so this is final point they link together, and then so the once the replication is done, so they can separate completely. So those are two separate daughter molecule. Okay. So now let's work on this question. One DNA is replicated, two brand new nuclear strands are produced. So I give you 5 seconds. So the correct answer is false. Okay, it's not two brand new. Okay, so this is semi-conservative. So one is old, another is new. Okay, so this is semi. Okay, it's not two brand new. Now let's talk about talk about DNA. DNA's function. Okay, DNA's function is for the transcription and the translation. Transcription means uh, to transfer the genetic information from DNA to RNA. Okay, to make a RNA molecule. Translation, that means read the information from RNA to make the protein. Okay. However, because we know we have the RNA virus. Okay, so RNA virus can convert RNA to another RNA. Okay, and then so RNA virus can use the RNA to for the translation. Okay, so that means they don't need the transcription. Okay, so they can use the RNA directly to make the protein. Okay, retrovirus actually so they also contain so they are also the RNA. However, so they need the DNA. So they need the transcription. Okay, so they can convert RNA to the DNA, and then so the DNA will get into the host nucleus, start the transcription. Okay, so this is for the DNA. So the RNA is also used to regulate the gene's functions. So they can regulate the DNA activities. Okay, so they can also completely shut down the transcription. Okay, so we can talk about this later. So DNA can make the RNA actually. So the DNA can make different RNA such as tRNA, RNA, and RNA. RNA is is used to make ribosome. Okay, TRNA is used to transport the amino acid. So only the mRNA contains the information to make the protein. So this is regulatory RNA such as microRNA, interference RNA, antisense RNA. So those RNAs actually can inhibit DNA either DNA transportation or inhibit the production of the messenger RNA. For example, microRNA can decrease can actually uh, inhibit the messenger RNA. Now let's talk about the transcription. So the initiation. Okay, initiation. So for the to start the transcription, so first we have to unzip the DNA double strand to expose the base sequence. Okay, so this can be done by the RNA polymerase. Okay, so they can unzip the DNA sequence, and then so this is the elongation. Okay, so so it can start the so this type is for the start making the RNA sequence. Okay, based on so they can use the one DNA strand as a template to make the RNA strand. So the reading direction is also from 5 to 3. You can see here. Okay, it's also from 5 to 3. So once the RNA strand is made and the process is done, so so it can be released to the nucleus into the nucleus, and so the unzipped DNA strand can be recorded together. So RNA is similar to the DNA, except RNA use U instead of C. Okay, instead of C. So RNA is actually the single strand molecule. So it exists in helical form because helical form can make RNA stable. Okay. So they can have secondary or tertiary structure. Okay. For example, for the tRNA has secondary structure. So the RNA has a tertiary. Okay, because ribosome has ribosome is made by two subunits, right? So the combination of two subunits actually has the tertiary structure. So I use U instead of timing. Okay, timing. Also, so I use ribose instead of deoxyribose. So the regulatory RNA are the microRNA, antisense RNA, and small interfering RNA. So those RNA can regulate the genetic function. So we also have the primer RNA. So this is work on both bacteria and eukaryotic cells and ribosome. Okay, ribosome. So this is enzyme made from RNA. Okay, and is to remove unneeded sequence from other RNA. So this is ribosome RNA. Okay, ribosome RNA. So actually, so there are two subunits of ribosome RNA. When they bind to each other, so they can make the mature ribosome, tRNA and messenger RNA. So the ribosome are the organized to make proteins. Okay. However, the ribosome is different in the precursors and eukaryotes. So the ribosome in precursors, mitochondria and chloroplast are the 70 and 70 size. Okay, so total is the 70s. However, so the two units are the 50 and 30s. 30s. Okay, so the combination of 50 and 30s is not 80, actually is 70. For the eukaryotic ribosome is total is 80. So two subunits is 60 and 40s. Okay. So they can bind to the 5 points end of the messenger RNA and read from 5 to 3 direction. Okay. So large subunits supply enzyme for the protein production. So we can see here in this diagram. Okay. So actually, so the small subunit bind with the messenger RNA, and the big subunit actually bind with the tRNA and supplies because tRNA is used for transportation of the amino acids. So they can, once they bind with the tRNA, so they can provide amino acids to make the polypeptide chain. So the codon, okay, so this actually, so in uh, genetic uh, the genetic information on the messenger RNA uses 3 nucleotides to dictate one amino acids. Okay, so 3 nucleotides stands for one amino acids. So this is called triplet code. Triplet code. So there are 64 different triplet code. Okay, and so all these 64 codes are used to some of these codes are used to represent 20 amino acids. Okay, so some amino acids may have more than one. They have several codes. Okay, so the meaning of for this is that so the cell can use this mechanism to prevent the mutation. Okay, even on the messenger RNA, even one nucleotide is muted. However, so the muted code may still stand for the same amino acids. Okay, so that will not change so that will not damage the protein production. Bubble, okay, bubble. So only the first 2 nucleotides are required for the coding. Okay, so the third actually is not important. So the change of the third doesn't change the coding. Okay. So this is another mechanism to prevent the mutation damage. Okay, so if the mutation happened on the third nucleotide, actually, so the meaning is the same. So this so the chapter8 still stands for the same amino acids. Okay, so we can see here. So this is all the codes. Okay, some codes stand for the so this two codes stand for the stopping meaning. Okay. Also this, so they can stop the transcription. So the others codes are used to stand for the different amino acids. So this is the diagram explains the interpretation. Okay. So this is triplets. Okay, triplets. The triplets in the DNA. So those information can be transcribed to the messenger RNA. Okay, messenger RNA. So the triplets can since the tRNA also has the triplets, so the triplets can so the triplets of the tRNA can match with the messenger RNA's triplets. So once they are matched together, so the amino acids can be uploaded and linked together. So this is the peptide chain. So here is the electron microscope view of the translation. So these are the small units and big units of ribosome. Okay, so these are the polypeptide chain. Okay, so this is the messenger RNA. So we can see here actually in just one. So in one mRNA string, so one mRNA string actually attached several ribosome. Okay, actually, so several translation can happen simultaneously together. Okay, so the efficiency of the translation actually is very high because one running actually can make several proteins. So we can see here first, so the small unit will bind with the messenger RNA, and then so the small units bind with big units. Big units will bind with tRNA. tRNA bring the amino acids to match with the code from a messenger RNA, and then so the amino acids can be linked together to form the polypeptides. So the translation in the bacteria, in the eukaryotes and prokaryotes are different. Okay. For example, in the precaryotes, so the start code always AUG. Okay. For the eukaryotes can be AUG. Okay. However, the code is still for different form of amino acids. Okay. Messenger RNA is the eukaryotes, so messenger RNA is only used for coding protein. In the precaryotes, so the messenger RNA can code for several genes. Okay, several genes. So the transcription translation, so since prokaryotes don't have the nucleus, so this happens in the cytoplasm. Okay. For the eukaryotes, so since eukaryotes has have the nucleus, so the transcription is in the nucleus. However, the translation is in the cytoplasm. Okay. So the genes. So bacteria use triplets to code for the protein. Okay. However, for the eukaryotes, since there are the introns doesn't code for the protein, so they have to be removed to make the messenger RNA. Okay. So this is the diagram shows transcription and translation. Okay. This is so this is shows the introns. So the introns have to be removed from the precursor messenger RNA. So all the extron will be linked together to make the mature messenger RNA. Now let's work on this question. Which of the following types of RNA contains the hairpin loop that is involved carrying amino acids to the ribosome during translation? I give you 5 seconds. Okay, so this is tRNA, okay, transfer RNA. So the genetic regulation of protein synthesis. So this is actually, so the translation is precisely regulated by the seers, okay? So once the seers need a certain type of protein, so they can start the translation. However, if they don't need those proteins, so they can shut down the production. So the regulation, so there are actually there are many types of regulation. Okay, so so here are some regulation. For example, antisense RNA, macroRNA, and rib switch. Now let's talk about the DNA level regulation. Okay, so the operon. Okay, operon. So the operon only exists in the bacteria and archaea. Okay, so this is in the prokaryotes. Okay. So the opera can be inducible or the repressible, okay? So it can also be classified as the catabolic operons, okay? So catabolic operons actually so for example, so if normally it is induced by the substrates of the enzymes, okay? So if the substrates of the enzyme so exist, so it can stimulate this operon and start the translation. Okay. So the suppressible operon, so it's known as anabolic enzyme. Okay, anabolic for the anabolic enzyme. So that means if those enzyme make the substrate, okay, this substrate actually will bind with the operon and shut down the regulation, shut down the translation. Okay, so so actually this is also another example of the negative feedback regulation. Okay, to prevent the substrates are making too much. Now let's talk about the lactose operon. Okay, lactose operon is also read as lac operon. Okay, lac means lactose. So this is the regulator. Okay, this is the regulator. So it contains. So it consists of the genes that code for the proteins. Okay, so the proteins can repress the operon. So this is called a repressor. And so there are two genetic fragment. Okay, one is the promoter, another is the operator. So the promoter will be recognized by the RNA polymerase. Okay, and the operator can be used can use as a switch to start or shut down the reading. Okay. So the structure locus actually are the sequence that code for different enzyme needed for the catabolism of the lactose. So we can use this. So the whole package of the lacto opera as a switch for certain genes. Okay, as we can see, so this is lacto opera. Okay, lacto opera. So this is promoter. Promoter can generate one protein, so they called a repressor. So the repressor can actually bind with the operator. Okay, so they can shut down this transcription. Okay, so the promoter actually can bind with RNA polymerase. Okay, however, since we have the repressor here, so even the RNA polymerase bind with the promoter. However, so the transcription is still inhibited. Okay. However, once we add lactose to the mediums, okay, so this exists in the bacteria. Okay. So once the bacteria eats the lactose, so lactose can bind with the repressor and release the repressor. Okay. So they can bind this part and release the repressor. If the repressor is released, so there's no inhibition. So the RNA polymerase can do its job. Okay, can initiate the transcription. Also can start the translation. So we can use actually the operon can be considered as the biological switch. So you can use this switch to control certain genes expression. So once the bacteria so this operon also can be used by the bacteria to adapt to the environment. Okay, so once the operon allows the gene expression, so the bacteria may change its fate. Okay, so we can know this. So this is considered as the phase variation. For example, the heritable phase. Okay, change. Okay, also. So they can change some trait affecting the surface of bacterial cells. For example, so the Neisseria, gonorrhea, okay, so they can produce attachment, okay, fimbriae, okay, so the Streptococcus pneumonia, so they can produce capsule, okay, capsule. So if the bacteria have the capsules, so they are very resistant to the antibiotics. They can also use the capsule to invade the host. Now let's work on this question. I'll give you 5 seconds. So the lac operon is so A is wrong, right? 5 now it's wrong. C is wrong, okay. 5 E is wrong. So a correct answer is D, okay. So it's wrong. So the operon will be switched on once the lactose level is high. Okay, lactose. So because this is for lactose. Now let's talk about the DNA recombination. Okay, recombination means to conjugate the one piece DNA with another DNA. Okay, because so the bacteria precursors don't have the real sexual reproduction. Okay, so they can use the unsexual reproduction to increase their numbers. However, so they need some process to recognize the environment and adapt the environment. Okay, so um, so so the bacteria has one specific event. Okay, so they can use this event to exchange their DNA information. So they can donate the DNA to another bacterium. Okay, so they can cause a new strain DNA generation. Okay, generated. So normally the plasmids are used to transfer the DNA information. Okay, for example, uh, so if one bacterium has the genes code for the drug resistance. Okay, so they can transfer these genes to another bacterium, so that is not resistant to the drug. Okay, so the recipient uh, so the bacterium that accepts the uh, donated DNA will express the resistance to the drug. Okay, so this is an example for the because when the bacterium accepts the donated DNA, so they can conjugate the donated DNA to their genome. Okay, and then so they can express the resistance to the drug. Okay, so this is one example for the recombination. Recombinant. Okay, recombinant is any organism that contains a gene that uh, originated in another organism. So, so the recombination is normally happened at the horizontal gene transfer. Okay, between the bacteria. So the another bacterium can get the new genes. Okay, so they can express the new trait of feathers. Okay, so the plasmids are widely used by the bacteria to transfer the genes. Okay, so plasmids are the uh, small circle DNA. Uh, so this circle DNA is replicated independently. Okay, independently, and so they can carry some genetic information for some specific trait. Okay. Plasmids are not necessary for survival. Okay, so if the bacteria doesn't have the plasmid, so they can still survive. Okay, so the plasmid DNA had to conjugated to the bacterium chromosome. Okay, to for the transcription and the translation, so that is the genetic information to be expressed because what we mean express is to make the protein. Okay, so the conjugation. So once so how the bacteria transfer their plasmids? So first, the two bacteria um, have to link together to exchange the contents inside of the cell. Okay, so normal actually, so they use the pillars. Okay, they use the pillars. So this is small hollow structure, so that can link two bacteria together to make the bridge between two bacteria to allow the exchange of the cellular contents. Okay, so this is conjugation. Another is the transformation. Transformation means the bacteria is actually phagocyte the DNA fragment. Okay, DNA fragment, so they can eat a DNA fragment, and then so they can, the DNA fragment can be conjugated with their chromosome. So the genetic information can be expressed and change the bacterial trait. Transduction means so the bacteria use the bacteriophage to transfer the small piece of DNA. Okay. So the example would be for the conjugation is the drug resistance of the resistance to the metal toxin. Okay. For the transformation, for example, the polysaccharide capture and all the cloning techniques. Transduction is so the example would be the toxin enzyme, so the fermentation and the drug resistance. Now let's talk about the conjugation. So the conjugation actually is more the genetic exchange. Okay. So in which the plasmid of the genetic material is transferred by donor to the recipient through the direct connection. So the direct connection actually is through the pilus. Okay, through the pilus. So this is through the pilus. So fertility F factor can allow the synthesis of conjugated pilus. Okay. And then recipient cell has the recognition site on surface for the pilus. So the pilus can find the recognition site and link these two cells together. So the donor cells has plasmid. Okay, so the recipient cells doesn't have this plasmid, so they need to exchange the plasmid. So once pilus is made from the donor cells, so they can attach to the recognition site of the recipient cells and drive the two cells together and make a tuner between these two cells. Okay, and so the cellular continents can change each other. For the gram-positive conjugation, actually, so this is an opening created between two adjacent cells. So this opening may be bigger. This means so this can be a bigger tuner between the two cells. So the fertility factor conjugation. So the donor cells actually had to make a copy of this fertility factor. Okay, and transmit this to the recipient factor. So the recipient cells. And then so the recipient cells can change its trait and turn into another donor cells, and then to make the pilus to transfer another to transfer the plasmid to another recipient cells. So high frequency recombination donors. So the plasmid becomes integrated to the donor chromosome. So one replication happened, so it began to transfer to the recipient cells. So we can see here. So this is fertility factor transfer. Okay, fertility factor transfer. So this is so they also use pillars to connect the two cells together, so to make tuners between the two cells. Okay, so the plasmids can be replicated to make a copy of the plasmid. So one copy can be transferred to the recipient cells, to let the recipient cells to have certain traits or features. Okay, so high frequency transfer. So that means the plasmid had to be conjugated to the chromosomes, bacterial chromosome. So once the chromosome is replicated, so the plasmid information can also be so the plasmid information can also be replicated. Okay, and then so the replicated copy will be transferred to the recipient cells to let so those recipients, so this plasmic fragment can be conjugated to the recipient cells chromosome. So the high frequency transfer actually use need the plasmid to conjugate with the bacterial chromosome. Okay. So the resistancy plasmid. So those plasmid can carry the genes to resistant antibiotics or certain toxin or metals. Okay, so they can be shared. Among the bacterium through the conjugation. So they can. So this plasmid can let the bacteria have the treated to resistance to the antibiotics, metals, or certain toxins. Okay. Now let's talk about the transformation. Transformation means the bacteria phagocyte the DNA fragment. Okay. So the cells, for example, if a bacteria is died, okay, so the bacteria can be lysis, okay, so the died bacteria can be break down. So all the cellular contents can be break down. So even the chromosome can be break down to the small piece. Okay, and then so the other cell, so another live bacteria can eat the small piece of the chromosome fragment. Okay, so this is the transformation. So this is another method that the bacteria to use to recognize the environment and adapt to the environment. Because when the bacteria eats the chromosome fragment, so the chromosome fragment can be conjugated to its chromosome, okay? So they can have more genetic information. So the bacteria that can eat the chromosome fragment is called a competent seer, okay? Competent. So they have this capacity, so this is called competent. So this means for the transformation, so the donor seer and the recipient seer don't have to connect together, okay? So they can live independently. However, if the recipient seer has the capacity to eat a certain genetic fragment, so they can do that. So they can they are called competent seer, okay? So the transformation is also used by the biological technology, okay? So we can use this technology to change some bacteria to let those bacteria to produce some substrate to produce some substance that we need, okay? For example, first we can make a plasmid. So the plasmid contains the genetic information that we need for produce certain substance, and then we inject the plasmid to the media. So the cells can eat the medium, can eat the plasmid and produce the substance. So the process that we use the plasmid to transfer the genetic information to the competent seer is called transfection. Okay, it's called transfection. So we can use this to change the trait of the yeast, plant cells, and even the animal cells. Transduction, okay, transduction. Transduction actually, so this is another method to transfer the genetic fragment. Okay, however, this method needs the bacteriophage. Okay, need a bacteriophage. So we use the bacteriophage as the carrier to transfer the genetic information. So we have two types of transduction. One is the generalized transduction. Okay, generalized transduction. So as what we can see in this diagram, so actually, so the genetic fragment so we can use the bacteriophage to transfer the a piece of genetic fragment to the cells. So this fragment will be conjugated to the bacteria chromosome, and then so the bacterial chromosome can make can duplicate can replicate this genetic fragment, can also make the genetic materials for the bacteriophage, and then so it will make more and more new bacteriophage. All this bacteriophage actually contains the genetic fragment that we need. Okay, so the new bacteriophage can also attack the other bacterial cells, and then as those bacterial cells to make more and more these genetic materials, the fragments. Okay, so this is a method to amplify the genetic fragment so that we need for certain production of the substance. So now let's talk about the special transduction. So as what we can see, so this is specialized transduction. Specialized transduction actually. So this is a highly specific part of the host genome. Okay, it's regulated, incorporated to the virus. So first, so the prophage DNA is separated from the bacterial chromosome and carry in a small segment host genes. And during lytic cycle, so this specific viral host genes can be incorporated to the viral particle and carried to another bacteria. So it's like in this diagram, okay? Now let's talk about the transposons, okay? Transposons. So the so the concept is jumping genes, okay? Jumping genes. So this is transposable elements. So it can shift from one part to another part, okay? So it can be transferred from chromosome to plasmid or vice versa, okay? And from cell to another cells, okay? So they can jump to so the replication jumped from one location to another location. So it involved in the change in trait, such as pigmentation and colony morphology, okay, replacement of damaged DNA. For example, as what we can see in this diagram. So this is transposon, okay, transposon can be either changed, so the transposon can change their location on the chromosome, okay. So they can also be replicated and they can also be replicated several pieces. So those so these multiple pieces can be conjugated to the chromosome as well. Okay, so we have more transposon fragment in the chromosome. So the transposon can be also transferred to the plasmid. Okay, and then to so the plasmid can transfer the transposon to another bacteria. Now let's work on this question. Which of the following mechanism of horizontal transfer involves the transfer of bacterial DNA through the bacteriophage? So I gave you 5 seconds. So the correct answer is D, transduction. Transduction needs the bacteriophage, okay? Transformation is bacteria phagocyte the genetic fragment. Now let's talk about the mutation, okay? Mutation means the genetic change, okay, genetic change. So any change to the nucleotide sequence will be considered as a mutation. Okay. So the mutation actually, since mutation is a change of the genotype, so the genotype change actually can cause the change of the phenotype. Okay. So that's why the mutation can cause the change of the trait to the living organism. So the change can be either harmful or the beneficial. Okay. So we have the different concepts to distinguish the mutation. So the Y type actually is natural unchanged characteristics. Okay, mutant strain actually that means the change of the nucleotide has happened. Okay, so so it can cause the change of the trait, okay, such as the morphology change, uh, nutritional characteristics change, okay, so the control mechanism change or the resistance to certain chemicals, temperatures, any type of enzyme functions, okay. So those all can also be considered as mutant trait. So the spontaneous mutation, so this randomly change during the DNA replication, okay? So this is actually, so the so the effect of the spontaneous mutation is mild, okay? So it can be corrected by the DNA polymerase very easily. Induced mutation actually is induced by certain conditions such as UV light, X-rays, okay, some chemicals. So those conditions can damage the DNA. Now let's classify the mutation. So first is the point mutation. Point mutation means the addition or deletion or change of the base. Okay, normally, so this is just one base change. So the missense. So the missense means the change of the code actually result in the change of the amino acids. Okay. So it can cause the replacement of amino acids. So that can be resulting another non-functional protein. Okay, because one amino acid is changed, so the protein may lose its function of faulty protein. Okay, or produce a protein that has the function completely different. Okay, or so no change because so even the so even so even the mutation of the code, however, doesn't change the even the change of amino acid, however, so the new protein, however, still have the same functions. So so nonsense mutation, that means so once we change those code, those triplets, okay, so the triplets um, becomes to a stop codon. So that means the translation is stopped. So so the so the protein will not be produced anymore. Silent mutation. Silent mutation means even the base is changed, however, so it doesn't change the amino acids. So that means there is no effect on the mutation. Back mutation means the mutation actually even the mutation happens, however, so the base is not changed. For example, UAA is mutated to UAG. Okay, UAG is continued to mutated to the UAA. So the final mutation is still the orange base. Okay, so this is back mutation. Fragment shift mutation. So that means one or more base are inserted or deleted. Okay, so this means one fragment is inserted or deleted. Okay, so this is a huge mutation. So it can change since a fragment of bases, so that can stand for a different meaning for the genetic sequence. So it can change the reading frame of the messenger RNA. So they can result in non-functioning protein. So this is the table shows the mutation. Okay, mutation. So this is missense. This is silence. So even the base is changed, however, so there's no effect. So let's talk about the single nucleotide polymorphism. So this is also called SMP. So this means only a single nucleotide is alternated. So it passed on genetically. So the identification is critical to the field of personalized medicine. For example, in the thrombophilia is one type of clotting disorder. So in this disease, a point mutation happened for clotting factor causes arginine to become glutamine. Okay. So the repairs of the mutation. Okay, repairs of the mutation. Since the DNA has its capacity to repair the mutation, for example, the polymerase 3 can correct the mistakes during the replication. So this is the capacity that cells have to prevent the mutation. So the CR also has the other system to correct the mutation, such as the CS has photodays. Okay, for this can repair the UV caused mutation. So, however, this repairment those enzymes can only repair the small numbers mutations. So if the mutation causes so there are too many or too large mutations, so those mutations cannot be repaired by the enzymes. So those mutations will cause trouble. For the repairment, so some enzyme will cut the mutated sequence. Okay, cut the mutant and replace with the normal sequence. Okay, so this is excision repair. So this enzyme will remove a defect base. So the remaining gap will be filled by the DNA polymerase 1 ligase. So once significant mutation happened, so also so those mutants is not repaired by the enzymes by the cells. So those mutants will be permanent, so they can be transferred to the offspring. Okay. So most of the mutation actually can cause trouble to the living organism. However, a small portion of the mutation can let the living organism get benefit. Okay. So actually, during the optimal environment, so the so the mutation actually is so the mutation is very rare. Okay. However, so even the small percentage of mutation can let the living organism has extra has a different trait that the living organism has the additional capacity to adapt the new environment. For example, those acquiring drug resistance can let the bacteria has the capacity to live in the environment with antibiotics, with the drug. Now let's work on this question. Which of the following type of mutations will have the most devastation effect on cells? So I give you 5 seconds. So the correct answer is B, fragment shift, since this is relative huge amount a base change, so it can cause significant effect on the mutation. Now let's talk about the DNA technology, genetic technology. So first, let's talk about the restriction endonucleosis. So this is enzyme can cut the DNA. Okay, can cut the DNA. So actually, many bacteria have this enzyme. So first of all, so bacteria use this enzyme to fight against the invasion. Okay, so they can use this enzyme to cut the foreign DNA to prevent the invasion. Okay, so can protect the bacteria against the incompatible DNA of the bacteriophage or plasmid. So they can prevent, for example, the viral DNA conjugation to their chromosomes. Now, so we can use this enzyme in the biotechnology factories to cut DNA and to generate a new bacteria to produce to make the substance that we want. Okay, so this is necessary for the recombination DNA technology. So how does the restriction endonucleases cut the DNA? Because they can recognize and cut at the palindromes. Okay, palindromes. So once the enzyme cut the DNA, so they can leave the short tail called sticky ends. Sticky ends. So once they cut the DNA, so they can cause the leaving overhangs on each end, and then so we can attach, so we can generate another DNA fragments with those overhangs to link to link our DNA fragment with the plasmid. Okay. So the restriction in the nucleus, once this enzyme cut the DNA, so the piece DNA that cut, so this is called restriction fragment. Restriction fragment length polymorphism, so that's a difference in the cutting pattern of the specific enzymes. Okay, specific enzymes. For example, so this is the palindrome. Okay, palindrome actually are the exactly same base sequence if we read from the 5 to 3 direction. Okay, for example, this is 5 to 3 direction. So if we read this sequence, actually is same from this one. This is read from this. Okay, from 5 to 3, 5 to 3. Actually, so this is the palindrome. Okay, so the restriction endonuclease will recognize this specific genetic fragment and cut this fragment. So this is when we cut, so this is called a restriction fragment. So we can have the specific overhand terminal, okay, and then we can generate another genetic fragment, DNA fragment that contains the overhands. So we can link our DNA fragment with this plasmid together, so we can make a new plasmid. So the ligase means to link the DNA fragment to link to separate the DNA fragment. Okay, so this is the function of the ligase. So they can sear the sticky end together. Okay, sticky end together. Reverse transcriptase. So they can make the DNA based on the RNA templates. So if the microorganism have this enzyme. For example, the virus, one type of virus is called a reverse transcript virus. So those viruses also means actually those viruses are the RNA virus. So they have the RNA. However, they also carry this type of enzyme. Once they invade the host, so they can use this enzyme to make the DNA based on their RNA template. Okay. For example, the HIV virus is type of reverse transcript virus. Okay. Complementary DNA. So actually, it's made from the mRNA, tRNA, or RNA. Okay, so this actually is the final product of the reverse transcriptase. Okay. Polymerase chain reaction. So this is a very significant event. Okay. So this is the technique allow the people to amplify the DNA products. Also, we can use this to purify the DNA. So now it's widely used to diagnose the diseases in the clinic. Okay, to detect the cancer, can detect certain infections such as viral infections. We use this a lot. So the process, even the process needs about 2 hours. However, the process is very fast. Okay, to amplify tons copies of the DNA. So the PCR needs the primers. So this is a short fragment DNA. It's about 15 to 30 base long. Okay, so we can use this primer to localize the DNA fragment to initiate the amplification. And then, so the PCR reagent contains the DNA polymerase. So actually, those are the enzymes that can work at a very high temperature. So those enzymes are isolated from the bacteria in from the bacteria lives in the very hot water. Okay. So PCR actually, so we have the devices to make about 30 cycles. Okay, and so each cycle has the high temperature and low temperature. Okay, so so the devices can perform the cycle one by one to amplify the DNA fragment. As what we can see in this diagram, so this is the DNA fragment. Okay, so first, so the PCR needs 95 degree. Okay, 95 degree. So at this degree, so the hydrogen bond between the nucleotide will be broken. So the substrate of the DNA will be unzipped, and then so the PCR decrease the temperature to the 70 degree, and then to allow the primer attached to the DNA fragment to localize the replication to localize the to set up the location for the replication, and then so the DNA polymerase can start the replication from the primers. So once the so this is called extension. Okay, once the replication is done, so we have two. So we have two. Actually, we have two copy of the DNA. Okay. Once the replication is done, so the PCR devices can go to the second cycle, so can heat the solution again, heat the solution to the 90 degree to allow these two to let these two copy DNA to separate again. Okay, to separate again. So this is called denaturation. Okay, denaturation to separate again, and then decrease the temperature to the 50 degree to allow the primer attached to the denatured DNA strand, and then raise the temperature to the 70 degree to allow the DNA polymerase to replicate the DNA fragments. So the PCR devices can repeat this cycle again and again until to 30 cycles, and then so we have tons of replicated DNA fragment. Once we get the amplified DNA products, so we can run the electrophoresis to detect the amplification and purify the DNA product that we need. So the electrophoresis actually is the agarose gel. So we made agarose gel, and then so we dump our so we can detect our DNA fragment. How do we detect our DNA fragment? So we can use restriction. So we can use restriction endonucleases to cut our DNA fragment, to cut different size fragment. Okay. So based on the cutting set, so we can design the size of the cut DNA fragment, and then we run the electrophoresis on the agarose gel. So this is so we can use, so this is the ladder. Okay, so the ladder shows us the different size of the so the ladder shows the size of the different DNA fragments. So based on the ladders, we can know if the DNA that we amplified is what we need. DNA sequencing, that means, so this technology allow us to know the base, so the information of base sequence on the chromosome. So this technology, so first we have to use the restriction endonucleases to cut the genome, okay, the chromosome, because the chromosome actually is a huge long DNA strand, okay. So we have to cut those long DNA strands to many small size DNA fragments, and then we run the electrophoresis to separate those DNA fragments. So we can pick the different. Also, we can pick the DNA fragment and purify the DNA fragment, and then we insert the DNA fragment to the plasmid and do the cloning. Then we let the competent cells to eat the plasmid and amplify the plasmid, because those plasmid contains the DNA fragment. And then we can use the sequencing machine to sequencing the DNA fragment on the plasmid. So the sequencing machine will give us the base information of the DNA fragment. So the computer will generate a report on the sequencing data. So once we finish the sequencing on all the DNA fragments, so we can use the computer software to link those DNA sequence together, so we can get the DNA sequencing on, so we can get the sequencing information on one DNA from the chromosome. So as what we can see in this diagram, so first we cut the DNA into small pieces, okay, and then we insert the small piece to the plasmid, and then let the competent seer to eat those plasmid to amplify the plasmid. So once we have enough amount of plasmid, so we can use the sequencing machine. Sequencing machine actually is the PCR machine to sequence the DNA fragment. So the sequencing machine will generate the sequencing report, and then once we finish all these piece sequencing, and then we can use software to link those sequencing report together, so we have the final sequencing data of the DNA. Now let's talk about the recombinant DNA technology. So this is widely used for the biotechnology factories or lab. So we can use this technology to produce hormones, enzyme, and to produce vaccine. So the gene cloning, that means we can design genes and then or we can get a gene from bacteria, plants, or the animals. So how we get these genes? So we can use restriction endonucleases to cut these genes from the hosting cells, okay, and then isolate this gene. We can insert this gene to a vector. So normally it's a plasmid or the virus like a bacteriophage, okay. So the vector we can let the competent if we insert gene to the plasmid, we can let the competent seer to eat this plasmid and then amplify this plasmid. So once we have enough plasmids, we can use this plasmid to transfect the cells that we use for produce certain substance. For example, the yeast. Okay, so once the yeast is transfected with this plasmid, so they can conjugate the DNA fragment to their host chromosome. And then, so if we grow tons of yeast, so they can produce the substance that we need. So this is the diagram shows gene cloning. First, we get the DNA fragment that we need, and then we insert to the plasmid. So we can get competence to amplify the plasmid to huge amount, and then we can use this plasmid to infect the cells. Okay. So the cells, can either contain the plasmid or conjugated the DNA fragment to their chromosome. So then we can grow tons of these cells. For example, the yeast cells in the container. Okay, and then so those cells will produce will translate the DNA fragment to make the substance for us. So the omics means the studies. Okay, means the systemic study. So the genomics means the systemic study of the genes. Okay, of the genomes. Okay. Proteomics means the systemic study for the cluster of proteins and the protein's functions. Metagenomics, that means the study for all the genomes in the ecological niche. Metabolomics means the study for a cluster of chemicals present in a cells. Synthetic biology. That means so this is the study area and a subject that produce that make the living organism. Okay, for example, in 2010, so Greg Venter created a bacteria from from the from the chemicals. Okay, so this is a huge scientific discovery. So we can use this technology to generate a new therapy method for certain diseases. For example, the gene therapy. Okay, gene therapy, and because our human has the genetic diseases. Okay, so this is the inheritance disease. For example, maybe the patient doesn't, the patient are lack of certain genes, so we can insert the genes that, so we can insert the genes that those patients are lack to the patients. So, so the patient have these genes, so the patient will can get the treatment for their genetic diseases. For example, we can use the virus. Normally, we use the adenovirus to carry the genes to the patient's cells. Okay, so the patient's cells can have this gene, so they can get the treatment for their disease. So the CRISPR is a very new technology, can allow scientists to cut the DNA. For example, if the patient have had a small piece of harmful DNA, so we can use this technology to remove this piece of DNA to 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 treat the patient's disease. Now let's work on this question. Restriction endonucleosis is what? So I give you 5 seconds. So we can see A, cut DNA at palindrome. So that's correct. Leave sticky end at the end of DNA cut. Yeah, that's correct. Used by the bacteria to protect against invading bacteriophage. Correct. Important in introducing a gene into a vector in the genetic engineering. That's correct. So the correct answer is E. All the choices are correct. So this is appendix. So you guys can use this for your reviewing.